Okay, so it was requested that I did another video on cell division. I already did one on meiosis, so check that one out, but here is one on cell division if you need it. Once again, the audio on this isn't the best, but as time goes on, hopefully I can fix that a little bit for you. All right, so here's cell division. We start out in interphase. That's the very first part of cell division. Within interphase, which is the longest part of cell division, takes about 90% of the time. The cell is doing its normal cell processes, growing in volume, preparing to divide, and as it's preparing to divide, it replicates organelles for the new cell that it's going to be made, and it also replicates its DNA. So each of those chromosomes gets another section to it, which makes them replicated chromosomes. So this side of the chromosome here is the exact same copy as this side of the chromosome here. So this is called a replicated chromosome. We could also call it sister chromatids if we would like. All right, so after interphase, we are ready for mitosis, which is the division of the nucleus. This is where we get Met. So here we go. So we start out in prophase. In prophase, the centrioles are forming, and they have migrated to the two opposite sides of the cell called the poles. The spindle here is forming. The asters here are forming, and the asters will anchor this entire apparatus to the poles of the cell. You can see here that we no longer have a nucleus and that our replicated chromosomes are now just floating in the cytoplasm. Then we move into metaphase. Remember M for middle. This is where the chromosomes line up across the middle of the cell, known as the equator. And the spindle here is going to attach from both sides to the centromere of the chromosome. Remember, I like to refer to the spindle as the claw because it reminds me of a crane machine, because from metaphase going into anaphase, those sides of the chromosomes called chromatids are going to be separated from each other. They're going to move away from each other. Remember, anaphase, A for away. So this side of the chromosomes are going to be moved to this pole, and these will move to this pole. In telophase, you can see the cleavage furrow is starting to form because cytokinesis is happening as telophase is happening as well. You can see here we no longer have a spindle or centrioles. They have disappeared. But our nuclei are back, and they do form around the chromosomes that were separated to each side of the cell. These chromosomes will eventually unravel back into chromatin and relax. And then we have cytokinesis, where we have the division of the cytoplasm. When we get two cells, you can see they are identical to each other, and they are actually identical to the cell we started with, which I will show you now. So here is the entire process from beginning to end. Once again, interphase, this cell here that we start with should be identical to the cells we end with, and you can see that it definitely is. So I'm showing you interphase here in 2 steps, even though it is 1. Just wanted to show you how those chromosomes go from being 1 chromosome to a replicated chromosome. Then we go prophase, metaphase, anaphase, telophase, and cytokinesis. We end with 2 daughter cells that are diploid. They have the same amount of DNA as each other. They are identical, and they are identical to the parent cell over here that we started with. So much more simple than meiosis. Hopefully this helps. Let me know if you have any questions.